Welcome to the Explainer. Let's jump right in. If you're a focused learner trying to map out the highly technical but absolutely fascinating journey of exosomal RNA sequencing for biological research, you are in the exact right place. Today, we're going to trace the complete end-to-end journey of a sample. We'll follow it from the very moment it's collected all the way to extracting those publication-ready biological insights.
Okay, let's dive into this. For a really long time, we've basically viewed extracellular vesicles as cellular waste dumps, just trash. But now, we know they are so much more. Think of exosomes as cellular messages in a bottle. They're these tiny, lipid-enclosed packages that cells send out to communicate with each other. And hiding inside those incredibly small vesicles is an absolute treasure trove of RNA cargo. That cargo holds the secrets to intercellular communication, physiological changes, and complex biological responses. Unlocking those messages, though, well, that's a massive technical challenge.
So here is our roadmap for today. First, the exosomal RNA challenge. Second, sample isolation and extraction. Third, library construction strategies. Fourth, navigating the bioinformatics workflow. And finally, the genomics solution.
Section 1. The Exosomal RNA Challenge. Decoding cellular messages. Before we even pick up a pipette, what exactly are we dealing with here? Exosomes are cell-derived vesicles, and they are exceptionally small. We're talking just 30 to 150 nanometers in diameter. They travel through eukaryotic fluids, and their main job is transferring cargo, specifically RNA, from a donor cell to an acceptor cell. Because they mirror the authentic physiological state of the cell they came from, they act as crucial mediators of intercellular communication. That makes them a phenomenal target for non-clinical biological research.
Researchers are incredibly excited about this field, and for good reason. Exosomal RNA gives us massive advantages. For starters, you can sample non-invasively using cell culture supernatants or plasma. But the real magic? It's the robust stability. Look right here. The lipid vesicle membrane physically shields the RNA from degradation by RNA enzymes in the environment. So the RNA stays remarkably stable and incredibly diverse, giving us a real-time, highly accurate reflection of cellular physiological changes. It's packed with messenger RNA, microRNA, and long non-coding RNA.
Moving on to Section 2. Sample isolation and extraction. The first critical steps.
Knowing the RNA is in there is one thing, right? Getting it out cleanly is an entirely different story. There's really no one-size-fits-all method for isolating exosomes, and this brilliantly illustrates the trade-offs. If you're working with cell culture media and need the absolute highest purity, ultracentrifugation is your gold standard, even though it's pretty time-consuming. Got a large-scale project where scalability matters more than ultra-high purity? PG precipitation is super easy to implement. And if you're handling delicate plasma samples, Size Exclusion Chromatography is wonderfully gentle on the vesicles, though you will need an extra concentration step. You absolutely have to match your method to your sample.
Once you isolate those exosomes, extracting the RNA is literally like panning for microscopic gold. Exosomal RNA is incredibly scarce. We are talking nanogram amounts here. To guarantee downstream sequencing success, you have to hit exact extraction metrics. We're aiming for a total RNA yield of at least 20 nanograms, a concentration of 1 nanogram per microliter, a clean OD 260/280 ratio between 1.8 and 2.2, and ideally an RNA integrity number, or REN, of 6.5 or higher. Honestly, if you miss these targets, you're setting up your sequencing run to fail.
Section 3. Library Construction Strategies. Building the sequencing library.
Okay, assuming you successfully extracted high-quality RNA, your library strategy has to be perfectly matched to your target. So the crucial point is this: you cannot treat all exosomal RNA the same way. If you're chasing microRNAs, you have to use a small RNA workflow. That means selecting for tiny sizes, just 18 to 30 nucleotides, and doing short, 50 base pair single-end reads. But if you're looking at fragmented long non-coding RNAs or mRNAs, you need the total RNA workflow. That path requires removing ribosomal RNA and utilizing longer, 75 to 150 base pair paired-end reads to capture the whole fragmented landscape. You've got to choose wisely based on your biological question.
Section 4. Navigating the bioinformatics workflow. Making sense of the data.
Once the sequencing machine actually finishes its run, we leave the wet lab and enter the digital realm. Exosomal RNA data requires a highly specialized computational pipeline. First, we do quality control on the raw reads, then adapter trimming and filtering. This is absolutely vital because small RNA libraries have a ton of adapter dimers. Third, we map the reads to a reference genome, or miRNA database. Fourth, expression quantification. And finally, statistical normalization. Getting from messy raw reads to a clean, reliable expression matrix takes precision at every single step.
To show you just how incredibly powerful this workflow is in the real world, check out this fascinating case study. Researchers used exosomal RNA-Seq to investigate dietary impacts. Get this, they actually isolated exosomes from cooked pork, fed them to mice, and proved that the specific microRNAs inside those pork-derived exosomes were absorbed by the mice. These dietary cellular messages directly altered hepatic lipid metabolism and caused metabolic disruptions in the mice. No way, right? It's just a mind-blowing demonstration of tracking cross-species intercellular communication using this exact technology.
But as amazing as those results can be, the path is basically covered in traps. You have to be incredibly careful during data interpretation. The biggest pitfalls? Over-interpreting low-abundance transcripts, where small fold changes might just be background noise. Ignoring cell-free RNA contamination that will completely drown out your true exosome signals. Misattributing tiny degraded mRNA fragments as fully functional full-length transcripts. And maybe most dangerously, failing to use proper statistical normalization. Fall into any of these traps and your entire experiment's data could be ruined.
As the quote goes, biological insight depends on correct interpretation. You could have the most expensive sequencing run in the world, but if your interpretation is flawed, the biological insight is completely lost. Because this complex workflow is so full of technical hurdles and analytical pitfalls, trying to manage the whole shebang yourself can be totally overwhelming, and that is exactly why partnering with an expert is usually the most reliable path forward.
Which brings us to Section 5, the CD Genomics solution. From DIY to done for you. Honestly, it's time to move from a DIY struggle to a done-for-you masterpiece. CG Genomics offers a complete end-to-end service designed specifically to handle the unique quirks of exosomal RNA. They specialize in the exact problem we've been talking about: low-input, vesicle-derived RNA. They can isolate from virtually any biofluid, perfectly optimize the library construction for either your small RNA or total RNA needs, and best of all, they provide custom bioinformatics and pathway enrichment analysis. They literally take over the heavy lifting of that entire computational pipeline we just reviewed.
Of course, to give you the best data, they demand the best starting material. By adhering to strict sample submission guidelines, CG Genomics guarantees high-quality inputs. They generally need at least 100 nanograms of total RNA at a concentration of 20 nanograms per microliter or 15 milliliters of cell supernatants. The samples absolutely must be strictly DNA-free and stored safely in nucleus-free water. By enforcing these rules up front, they ensure the sequencing process has the absolute highest quality material to work with from day one.
Now, what's really interesting about this slide is the sheer firepower CD Genomics brings to the table. They offer incredibly flexible, high-throughput sequencing strategies. Whether you need an Illumina HiSeq 4000, a NextSeq 500, or the MGI DNB Seq series, they have the platforms optimized to deliver massive data volumes, typically around 10 gigabases of deep sequencing data per sample. And they can accommodate both single-end and paired-end read requirements effortlessly.
And the final output? It isn't just some massive, unreadable file of raw data. It's a fully contextualized, publication-ready package meant to accelerate your non-clinical biological research. Yes, you get the raw data and QC results, but you also get annotated expression matrices, stunning high-quality heat maps and volcano plots, intuitive functional enrichment visuals, and, this is huge, a detailed methodology section that is completely ready to be dropped right into your paper's supplementary materials.
So, as we wrap up this explainer, I want to leave you with a provocative thought. How will CD Genomics streamline your next exosome RNA-Seq experiment? The journey of a sample is undeniably complex, right? It's fraught with low-input challenges and bioinformatics hurdles, but you don't have to navigate it alone. By abandoning the DIY struggle and leveraging an optimized expert pipeline, you can bypass the pitfalls and focus on what really matters, unlocking your next major biological discovery. Thank you so much for joining me today, and keep exploring those cellular messages.